हम रिकॉर्डिंग स्टार्ट करते हैं। सना मसला पार्टिसिपेंट्स का वेटिंग में। एनीवे सो लेट्स स्टार्ट। आप सबको आवाज आ रही है मेरी?
जी सर आ रही
जी
है।
सर।
Okay, so there are four different ways. We have previously studied that the reader-writer complexes and different types of proteins, they can read the histone code, or there are other proteins which can modify the histone code. We have talked about the reader-writer complexes. अगर आपको नजर आ रहा हो पिछली slides हमारी, so we have studied this, that there are different types of reader-writer complexes. A gene regulatory protein that binds to any of the specific DNA regions, it can recruit certain other histone modifying enzymes like writers or readers, etc. And they can proceed with this modification on the chromatin. And they can either uh, uh, change the chromatin from euchromatin to heterochromatin and silence the genes, or they can revert it from heterochromatin back to euchromatin, and then they can initiate the transcription process. How do they do this? So we are going to study that how does this gene regulatory protein, so this is where it started, the modification was the first binding of the gene regulatory protein to the DNA. So we are going to, excuse me, my ये mask उतार लूं, आप लोगों को आवाज साफ आएगी। So the transcription starts from binding of the gene regulatory protein to the DNA. So how does this gene regulatory protein carry out modification of the chromatin? That's what we are going to study. And we're only talking about the activation of the gene. We will talk about the expression of the genes later. So today we are going to study how do these proteins, these activator proteins, we have previously studied what are activator proteins, the proteins that lead to the initiation of the transcription. How do they direct local alterations in the chromatin structure to stimulate transcription initiation? Means how do these activator proteins activate the transcription of the genes? We will just study the role of activator proteins, and we will see how do they initiate transcription of the genes by modifying the chromatin structure. So what happens here, we have some heterochromatin, and we see that, for example, this was a gene which is wrapped around the nucleosomes, and here we have the Tata box. The Tata box is embedded inside the chromatin, so it's not possible for the transcriptional machinery to access the Tata box. Transcription machinery means our general transcription factors and RNA polymerase, for example. So they cannot go into the into this chromatin and reach the Tata box to start the transcription. So what do we need to do? We need to make this Tata box, or this initial the transcription binding site, available to the transcription machinery. So what happens? First of all, a gene activator protein binds somewhere on the chromatin to a region which is accessible. So when this regulatory protein binds here, this gene activator protein, where do I get these lines? आप सब लोगों को आवाज आ रही है मेरी?
जी सर आ रही है।
ओके, किसी का कोई क्वेश्चन हो तो यू कैन आस्क मी। यहां तक समझ आ रही है आप लोगों को? हेलो?
जी सर
समझ आ रही है।
आ रही है, okay, that's good. So the gene activator protein binds here. What can this initial gene activator protein do? It can stimulate transcription in 4 different ways. So first, study the last one. What is the last one? This is what this gene regulatory protein can do. This can recruit a histone modifying enzyme. And what is a histone modifying enzyme we have previously discussed? These could be acetylases, which can histone acetyltransferases, HATs, which can carry out acetylation. And we know that acetylation relaxes chromatin, or it can also recruit the demethylases, which can remove the methyl group to relax the chromatin. So if the chromatin is relaxed, then this Tata box can become accessible to the transcription machinery. So we have studied these things in detail previously. How do these histone modifying enzymes can relax the chromatin? If you remember the previous slides, here. So how could these modifications relax the chromatin? So if you acetylate the histone tails, then there would be a negative charge on the histone tails, and this would repel the DNA or the nucleosomes from each other, and this would relax the chromatin. So these are our histone modifying enzymes, which are recruited by the gene regulatory protein. So this gene activated protein binds to the chromatin and recruits a histone modifying enzyme. And this histone modifying enzyme can modify the histones in such a way that the chromatin is relaxed. So this is one way how this gene activated protein can relax the chromatin so that Tata box or the transcription initiation site, they become accessible for the transcription machinery to bind to this region and activate this region. The other three methods, they are dependent on chromatin remodeling complexes. So these are complexes of proteins, and they can carry out different types of modifications to make this site accessible to the transcription machinery. How do they do this? So there are three different mechanisms. First one is nucleosome sliding. So a complex of protein binds to this particular region, for example, in this region. So the gene regulatory protein binds here, and this complex binds to this region. And by binding to this region, what it would do, it would slide these nucleosomes away from each other so that the Tata box becomes accessible for the transcription machinery. That's what we call nucleosome sliding. Second possibility is where chaperones play a role. So what do these chaperones do? These chaperone proteins can remove the histone proteins. So this is what we call histone removal. So histone proteins, we know that they stabilize the DNA. The DNA is wrapped around them, and when these histone proteins are removed, then obviously the DNA wrapped around them is liberated and becomes accessible to the transcription machinery. So this is the second mechanism how the gene activator proteins can make the Tata box accessible to the transcription machinery. And the third one is histone replacement. How does histone replacement take place? There are different types of histones. We call them histone variants. So what are these histone variants? Basically, they have a different type of modification. Let's assume that a histone here previously was methylated. And we know that methylation inhibits transcription. So what chaperone is going to do, this would bring in another histone which is acetylated. It will kick out the methylated histone from here and insert the acetylated histone here. So this is what we call histone replacement, that one type of histone is replaced with another type of histone. And we also know this is one thing which is modification, and we also have slightly different variants of the histone proteins. We have these four histone proteins: H2A, H2B, H3, and H4. We will talk about one example of another variant in this class. You will understand it better then. But what you need to understand at this moment is that one type of histone protein here out of these four can be replaced with another type of histone molecule. And this histone molecule would facilitate. आवाज आ रही है आप लोगों को?
जी सर आ रही है.
Okay.
जी सर आ रही है.
So one type of histone molecule or histone protein is replaced by another type of histone protein. So the new protein now facilitates the transcriptional activation. So these are four different mechanisms that the gene activator proteins can carry out, or they can follow in order to stimulate the transcription initiation. One is nucleosome sliding. Second one is histone removal, that the histones are removed, they're kicked out, and the DNA is liberated. And the third one is that the existing histones are replaced with another type of histone. And the newer histones, they facilitate the transcriptional activity. And the fourth mechanism is recruitment of the histone modifying enzymes. And what do these histone modifying enzymes do? They will carry out modifications. For example, acetylation. And acetylation would relax the chromatin, and the chromatin will become accessible for the transcription machinery. Clear to everyone? Any questions so far? किसी चीज की समझ ना आई हो?
No, sir.
Okay, clear to everyone?
Yes, समझ sir.
Okay, good. So
Yes, sir.
what happens, for example, in A, what we see, these are the same three mechanisms. We are not talking about the histone modification. We are talking about the remodeling. So the first thing, we have studied this remodeling, that these remodeling complexes can carry out three different processes. They can do histone sliding or nucleosome sliding. They can carry out histone removal, and they can carry out histone replacement. So these are the same things: nucleosome sliding, histone exchange, and nucleosome eviction. Eviction means removal. So these are three mechanisms. So these three mechanisms would stimulate the initiation of transcription, and that happens because of the binding of these remodeling factors to this heterochromatin. So what happens when this remodeling factor is removed? After removal of this remodeling factor, the chromatin has to be put back into this heterochromatin form. So it needs to be compacted again or condensed again. So that would be mediated again by the chaperones, histone proteins. So chaperones they would bring in the histone proteins and we'll put them back if in case where the nucleosome was removed. So if the nucleosomes were removed, they put back these nucleosomes or histone proteins, and then the DNA is wrapped around them, and this constitutes the nucleosomes back again. And the second thing they can do after they're released, if they had carried out this nucleosome sliding, so they put the nucleosomes back closer to each other, and thereby they restore the chromatin back to its pre-activation form. So this happens only when activation of the genes is needed. So if the activation of after the transcription is completed and it's not needed anymore, these remodeling factors, they are removed and they put this thing back into the pre-activation form. They put the chromatin back into pre-activation form. So two things are happening when these remodeling complexes or the histone modification enzymes, when they bind to the DNA, to the chromatin, they relax the chromatin so that the genes become accessible to the transcription machinery and transcription can be carried out. And after this transcription is not needed anymore, these remodeling complexes, they are removed from the chromatin. These enzymes are either removed or a different type of enzyme is recruited. For example, the enzymes which would carry out methylation, so that the DNA can be put back into its euchromatin form and the genes become silenced. Clear to everyone? Any questions so far? Any questions?
No, sir.
No, sir.
Okay. Should we proceed further? Okay, so these type of modifications, they can last for different times. They can these could be long-term modifications. These can be short-term modifications, and these can be modifications for medium medium time. So all of these lengths of time, they're dependent upon the type of modifications and the type of nucleosomes which have been exchanged. So different types of modifications would have a different impact on this gene activation. Some of them would activate the genes for a shorter period of time, some of them for midterm length, and some of them for a longer period of time, for such a long time that these modifications can even be passed on to the next generation. For example, if certain genes were activated, then this activation would last for a longer period of time, and this can be passed on to the next generation. But in certain cases, this is pretty short. There's, I mean, during the transcriptional activation, what happens, some types, a special type of modification is so short that, for example, if the chromatin was relaxed here, the RNA polymerase 2 binds here to the Tata box. When the RNA polymerase 2 binds here, RNA polymerase 2 with itself carries these code reader and writer complexes. So what do they do, these enzymes? So what are these reader writer complexes? These are basically histone modifying enzymes. So when the RNA polymerase binds here, only a small region like here is basically modified. So after binding of the RNA polymerase here, the RNA polymerase along with it carries some writer complexes. What do these writer complexes do? They carry out acetylation of the following histone proteins. And after acetylation, these histone proteins are modified and the DNA is relaxed. And the RNA polymerase initiates transcription. So the histones are removed, the DNA is liberated, and the RNA polymerase travels further along the along its transcriptional path, or along the DNA from 5' end to the 3' end. So, but as soon as the RNA polymerase moves away from this spot, so the DNA behind the RNA polymerase is wrapped around the histone proteins once again. So histone proteins are brought in and the DNA is wrapped around them. Why? Because the same RNA polymerase also carries some other different type of enzymes, for example, methyl transferases, which would immediately methylate the histone proteins, and the DNA will be put back into its condensed form. So not even an entire gene is relaxed, only a short portion of the DNA or of the gene is relaxed. The RNA polymerase keeps moving forward, making its way through the compact chromatin by modifying the histone proteins and removing them. It moves forward, and as soon as it moves forward, it also carries histone methyl transferases. So it recruits the new, the chaperones will bring in the new histone proteins, and the writer complexes which were carried along by the RNA polymerase, they would immediately methylate these histone proteins, and the DNA will be again condensed. So it is like RNA polymerase is moving in the forward direction, removing the histone proteins, making its way through for transcription, and then as soon as it moves forward, the new histone proteins are brought in, and then they're immediately methylated to put the chromatin back into condensed form. So this is a very short term modification कि RNA polymerase आगे बढ़ता जाएगा और उसके पीछे वाले जो हैं वो modify होते जाएंगे. So clear to everyone? Any questions so far?
No, sir.
Okay. समझ आ गई सबको?
जी सर.
Okay. I hope there's no one waiting. Sana Fatma joining लिखा हुआ है, but I'm not sure if she's still unable to join for whatever reasons. Anyways, let's move further. So we have seen that these histone modifications, they can influence the chromatin structure, these gene regulatory proteins. But even before these activator proteins can bind to the DNA and can carry out the modifications, so the region before this transcription site. So for example, this is the region. Sometimes what happens that this is the region that needs to be, this is the Tata box where the RNA polymerase needs to bind. Sometimes there's a nucleosome-free region already available so that the gene regulatory protein can bind to it. Or for example, this region. The region closer to the DNA, closer to the Tata box, or closer to the gene that needs to be transcribed. Sometimes this region is free of nucleosomes. So there are no nucleosomes in this region. if we look at this region, for example. So this would be a nucleosome-free region so that a gene regulating protein can bind to it. So we say that the the DNA is or the genome is poised to become activated. Already उसमें एक चीज रख दी गई है कि इसमें activation करनी है या इसकी activation होनी है. So, I'm not sure why the Chromatin. Okay, she has joined. Okay. So sometimes these regions are nucleosome-free regions, and how are they maintained as nucleosome-free regions? So generally, these nucleosome-free regions are flanked by a special type of histone variant. We call it H2AZ variant. So basically, we know that there are four types of histone proteins: H2A, H2B, H3, and H4. So generally, H3 and H4, they are towards the inside of the chromatin, and H2A and H2B are towards the outside of the chromatin, towards the exterior, towards the surface. So H2A, we just talked about the variants, that there are different variants of the histone of these histone proteins. Like a variant for H2A is H2AZ. So this is a variant because H2AZ can easily be modified, and it is easier to dissociate H2AZ from the chromatin as compared to H2A. So the region where a regulatory protein needs to bind, this has to be easily accessible for the regulatory protein. So this is the specific region where the activator protein has to bind. This region would be made accessible by the presence of histone variants. And for example, this region would be a nucleosome-free region so that the DNA is accessible, the DNA is free, and the activator protein can bind to it. So what are H2AZ variants? So as I have told you, that they're thought to be easily disassembled. They can easily be removed from the chromatin so that transcription can take place. We have two types of heterochromatins. One is known as a constitutive heterochromatin. I hope you understand the difference between the two. The compact form of the DNA is the heterochromatin, where the genes are silenced. It is not the relaxed chromatin, and the relaxed chromatin is known as euchromatin. Now, heterochromatin is of two types. One is constitutive heterochromatin, and the second one is facultative heterochromatin. So as the name indicates, we have previously discussed this in the, as far as gene expression is concerned, that constitutive is something which stays the same all the time. So a constitutive heterochromatin contains those genes which are permanently silenced. They do not need to be activated. And facultative heterochromatin contains the genes which need to be activated sometimes, and then they need to be silenced when the signal is gone. So what is this facultative heterochromatin? This facultative heterochromatin keeps switching between the euchromatin and the heterochromatin, so that when it switches between these two forms, when it goes from facultative heterochromatin to euchromatin, then the genes become accessible for the transcription machinery and they're expressed. And when it is condensed back into the heterochromatin form, they're silenced. So these regions generally contain these histone variants, and a very famous common example is H2AZ variant. So what happens, rather than H2A, so the green one here is H2AZ. So rather than H2A, the nucleosomes here, they contain a variant of H2A, that is H2AZ. Now, when H2AZ is here, and we have this euchromatin which was expressing itself, here it is written K4 methylation of H3 tail. So if I switch back to one of our previous slides where we were talking about the histone code, here's the histone code. We have studied that methylation of K4 led to gene expression, and methylation of so K means lysine. So methylation of lysine at fourth position, fourth lysine residue, This methylation of lysine here at position number 4 led to gene expression, while methylation of lysine at 27th position leads to silencing of the genes. So this is the same same gene, basically the same histone protein. It is the same region on H3. So if K4 is methylated, the genes are expressed, and if K27 is silenced, then the genes are if it is K27 is methylated, then genes are silenced. So it is the same example here that we are talking about. So you see, euchromatin, where K4 is methylated, it is in the form of euchromatin. And sorry, K27, I say lysine, where lysine 27 is methylated, this is heterochromatin, and where lysine 4 is methylated, this is euchromatin. So this is switching back and forth between euchromatin and heterochromatin. So it is easier to modify H2AZ as compared to H2A, and they can also be evicted, they can also be removed very easily. So another thing that they can easily be modified, so after H2AZ variant is inserted into the nucleosome rather than H2A. So if this H2A gets acylated, this will lead to transcriptional activation. And if this is ubiquitinated, this will lead to transcriptional repression. So these variants also play a specific role, a special role in the conversion of chromatin between the two forms, euchromatin and heterochromatin, and they also play a role in transcriptional activation and transcriptional repression. And they're very important in regions where regulatory proteins bind. So we are also running out of time. We have 10 minutes left. I think we can wind it up. So they can switch back and forth between these two forms, and these histone variants, H2AZ, they're generally found in regions which are closer to the regions where regulatory proteins bind. For example, in this region. So this would be a nucleosome-free region, and it would be flanked by these H2A variant histones. Why? Because when the gene regulatory protein binds here, it can easily modify the nearby nearby nucleosomes. Why? Because these nucleosomes, the nearby nucleosomes, this one and this one, for example, they are carrying the H2AZ variant, and we have studied that it is easier to modify and it is easier to remove or kick out these H2AZ variants, and removal of them would help the activator protein to initiate the transcriptional activation. Okay, thank you very much. I think that is sufficient for today's class. If you have any questions, please. If anyone has any questions, please. आप लोगों को आवाज आ रही है मेरी?
जी सर, आवाज आ रही है। एक दफा रिकॉर्डेड लेक्चर सुन के फिर कोई क्वेश्चन होगा तो सर वो गूगल क्लासरूम पे पूछ लेंगे।
So आप लोगों को समझ आ गई है? Everybody got what we talked about?
जी सर, समझ आई है।
जी सर, समझ आई।
Yes सर, आ गई है।
Okay, that's good. I have recorded the lecture. I will post this recorded lecture. I hope audio भी clear रही आज की क्लास में.
Yes सर, ठीक थी आज। जी सर, आज तो अच्छी थी।
Okay, that's good. So we talked about we have 7 minutes left. I would just quickly go through it once again. The gene activator protein binds to its regulatory site. It can recruit chromatin remodeling complexes. What they can do, they can carry out the nucleosome sliding, or they can remove the histone proteins, or they can carry out histone replacement in order for the Tata box to become accessible to the transcription machinery. Or the other thing they can do, they can recruit histone modifying enzymes. For example, histone acetyl transferases, they can carry out acetylation of the histone proteins, and this would relax the chromatin. and the DNA would become accessible to the transcriptional machinery, and this will lead to the transcriptional activation of the genes. And then we studied what happens when these remodeling factors are removed. When they're removed, then so the chromatin is put back into its, the euchromatin is put back into the heterochromatin form. The sliding is I don't know, they are put back closer. The nucleosomes are put back closer to each other, or if there was nucleosome exchange, again the different type or a variant of the histone is added, or if there was nucleosome eviction, then the histone proteins are added back by the chaperone proteins in order to assemble the nucleosome once again. Okay, and then we also talked about the different time points that different types of modifications can have different impacts. They can be so short, sometimes they can be so short that the RNA polymerase 2, which carries along itself specific types of histone modification enzymes, that it keeps moving through the chromatin and it keeps modifying the histones next to it, and it keeps removing them so that the DNA is liberated. It keeps translating the DNA, transcribing the DNA, and as soon as the RNA polymerase moves forward, the new histones are brought in and they are immediately modified by the methyltransferases, histone methyltransferases, which are also carried along by the RNA polymerase. It keeps moving in along the chromatin, keeps removing the histone proteins, keeps transcribing, and then after it moves further, immediately behind the RNA polymerase, the histone proteins are inserted back and the chromatin is condensed. So this is basically done to prevent the I don't know, unintended transcription of the genes, so that it does not take place without any reasons. And sometimes it can be so long that it can be passed on to the next generation. And then we talked about the histone variant of H2A, which was H2AZ, and H2AZ is generally found flanking the region where the initial regulatory protein or the activator protein needs to bind. For example, this one and this one. So these two histone proteins can carry, these two nucleosomes can carry a variant of H2A, that is H2AZ. Why? Because it is easier to modify this H2AZ and it is easier to remove it. So it facilitates the initial relaxation of the chromatin. And we talked about the two forms of the chromatin, heterochromatin. One is constitutive heterochromatin. So this contains those genes which are kept silent for the entire time. And there's facultative heterochromatin. The facultative heterochromatin contains the genes which need to be turned on time to time. So it keeps switching between these two forms, euchromatin and heterochromatin. And we have also talked of the histone code that we studied previously, that if the lysine 4 at H3 tail is methylated, we had previously studied that this leads to the gene expression, and if histone 27 is methylated, that leads to the heterochromatin formation. And this is why it happens. You have studied this in details now. So if acetylation takes place, transcriptional activation, and if ubiquitination or methylation takes place, that leads to transcriptional repression. I think that's sufficient for today. So if anybody has got any questions, otherwise we can wind it up. Okay, any questions?
सर अभी क्वेश्चन याद नहीं आ रहे.
Okay, thank you very much. I think
thank you.
It's over. Thank you very much for your participation. Yeah. Okay, Allah Hafiz.